here's protein and peptide hormones. All these guys, I've got a box around PTH and calcitonin. That's what we're going to talk about next. And then here's the steroid hormones. Look how they spelled estrogen. So what country do you think this table came from? Probably somewhere in Europe, right?
Yeah.
They spell it weird. Amino acid derivatives, thyroid hormones, we just talked about those. And then fatty acid derivatives, these guys are all associated with eicosanoids, but we're not going to have time to cover those. So. All right, so here's the thyroid gland. We talked about how you make thyroid hormone, but buried within the thyroid gland are parathyroid glands. And I think it was maybe in the 1800s, the surgeons, if you had thyroid cancer, they would cut out parts of your thyroid, and some people would live and some people would die from that operation. And then they found out that if they were cutting out these parathyroid glands, then those people all died. Because parathyroid glands regulate calcium, and calcium is super important in maintaining homeostasis. So here's your pharynx. It's posterior view of the pharynx, so you look from behind. Here's the thyroid gland. Here's the other part of the thyroid gland, kind of wraps around the esophagus and trachea. And then here's your parathyroid glands. So we talked about the thyroid gland already. It has follicular cells. That's where you make thyroid hormone. The thyroid gland also has these cells called parafollicular C cells. They make calcitonin hormone. And then buried within the thyroid gland is the parathyroid gland. And there's 4 of them. So 1, 2, 3, 4 parathyroids. And within the parathyroid gland, you have these chief cells, and they make PTH, parathyroid hormone. So we've got 3 cell types and 3 hormones you got to keep track of. So thyroid gland makes thyroid hormone. We just talked about that. The follicular cells in the thyroid gland make thyroid hormone. The parafollicular cells, parafollicular C cells, also in the thyroid gland, make calcitonin. And parathyroid gland, this is the easy one, parathyroid gland makes parathyroid hormone, or PTH. Where do you think you would find these parafollicular C cells?
Next to.
Yeah, around the follicle cells. Yeah, exactly. And we'll look at this later on in the microscope today. So here's a cross section of the thyroid gland. Here's the follicular cells. Can you see all these kind of dark blue things, squares? Where's a good place to see? Right here. Here's the nucleus. That's a nucleus of a parafollicular cell. These are all nuclei. So this would be all follicle cells. And what would all this junk be in the colloid? Yep. And then around the follicles would be these parafollicular cells. So here's parafollicular cells. And you find a bunch of parafollicular cells here. You guys remember your histology, or does that just look like a big mess right now?
Big mess.
Anyone?
How do you tell the difference between the follicle and then the parafollicle?
How do you tell the difference? Here's what I do. I find the follicle. So this whole big brown thing is a follicle, right? And then I look at the periphery, and all these cells that are lining the follicle are the follicular cells. And then any other cell type, like out here between, so let me back up. Here's a follicle, right? Here's a follicle. If you are between follicles, you are parafollicular. And then I just look for that blue blob, that's the nucleus. So all these cells are going to regulate calcium in the blood, which that range, that homeostatic range is about 9.2 to 10.4 milligrams per DL. What's DL?
Deciliter.
Deciliter. Yeah, good. All right, here's my drawing. I just think it's so brilliant. I hope you can see it. Looks like it's a little washed out, but here you are. You're all smiley. You've got your thyroid gland. Looks like a bow tie. And then buried within the thyroid gland are these parathyroid glands. And they have chief cells inside. So the parathyroid gland, it's got to be made of a cell type. What's its name? Chief cells. Chief cells and parathyroid sense low calcium in the blood. So that means there's capillaries running through the parathyroid gland, and they've got little meters on them. The cells, chief cells, they can sense when calcium is low. When calcium is low in the blood, what do you want to do to calcium? Via negative feedback, you want to raise calcium back up. So this is long, drawn-out process how to do that. So low blood calcium, chief cells secrete PTH. PTH travels all over the blood and binds to osteoblasts, clasts, and sites. Where is that?
Bone.
In the bone. Why would PTH, how is PTH going to help raise the calcium level in your blood? Because, yeah, your bones are made of calcium and other things, but a lot of calcium. So, what you want to do is you want to get your bone to release calcium. So, what are you going to tell the osteoblast to do?
Turn on.
Stop. It's gotta stop
I
building.
heard stop. You're right. So the osteoblasts build bone. That means they take calcium out of the blood to build bone. So, hey, osteoblasts, slow down. We want to stop your making bone, because when you make bone, you pull calcium out of the blood. So inhibit osteoblasts. What do you want to do to the osteoclasts?
Tell them to start tearing down.
Osteoclasts break down bone. So PTH tells osteoclasts, break down bone. When you break down bone, calcium is released into the blood. That brings your blood calcium levels back up. And osteocytes, we don't really care. They're not, I mean, they bind it, but they do something that isn't as easy to think about as an osteoblast or an osteoclast. So in addition to this, so you get calcium from the bone, in addition to this, PTH is going to go to the kidneys. How can the kidneys help bring calcium back up? Yeah, don't urinate calcium out. Hold on to calcium in the kidneys instead of I know everybody wants, it's so hard for me to say the word, can you say it? Pee? Just so, did I ever tell some of you guys that story? How I hate the word pee? Yeah, just like I was brought up in a family where only, how do I say this without sounding snobby, but yeah, you just, if you were of a certain type of person, you didn't say it, pee. You didn't say I have to pee. But now everybody says it, right? But while I was brought up, it's like, oh, I have to go to the restroom. I have to go to the lavatory. But I should just say PTH binds to the kidney cells so you don't pee out calcium. That's the easiest way to say it. But I'm having a PTSD right now, just even saying that. So I just had to let you know. So I'm going to try to say urinate, and you can substitute pee in your mind. But don't urinate out the calcium. Hold on to it. Keep it in the blood. All right. What's also going on at the same time? Here's the sun, UV radiation. UV radiation tells your keratinocytes, remember those from 201?
Mm-hmm.
Tells your keratinocytes to start making calcitriol. Calcitriol is another hormone. So in this cartoon we have two hormones, parathyroid hormone and calcitriol hormone. Calcitriol is also called vitamin D. Vitamin D, yeah. So calcitriol is really, that's hard to make that hormone. So you start making it in the keratinocytes, then you go to the liver, the hepatocytes continue making calcitriol. Then you go to the kidneys, and now you're calcitriol, but you're not fully grown calcitriol in the kidneys until PTH is there. Then when PTH is present, PTH is going to finish calcitriol synthesis. What can calcitriol do as a hormone? Calcitriol can go to the bone. So in addition to PTH, calcitriol can go to the bone. And it says decrease bone reabsorption. It says bone reabsorption of calcium. It's going to increase bone resorption of calcium. What's bone resorption? Is that breaking the bone down or building it up? It's breaking it down. So calcitriol, in addition to PTH, is also going to increase bone resorption. And then calcitriol will increase GI tract absorption of calcium. So anything you eat with calcium, calcitriol is going to say, hey, hold on to that calcium. Put it in the blood. Because remember, what was the original stimulus? Low blood calcium, aka hypocalcemia. And then also there's, you can go to the kidney, calcitriol will say hold on, reabsorb calcium. So reabsorb calcium back into the blood. And don't let calcium be urinated out in the urine. So that's the hypocalcemic story. So you can have low blood calcium. You can also have high blood calcium. What's that called? Hypercalcemia. And there's a mechanism for that too. So for hypercalcemia, you have your parafollicular C cells. Where do they live? Around the follicle of the thyroid gland. Yeah, around the follicle of the thyroid gland. They have little calcium meters on them too. They sense when calcium is high. When calcium is high, they secrete calcitonin. When calcium is high, what do you want to do to blood calcium? If blood calcium is too high, you want to bring it back down. So you want to, under the influence of calcitonin, you want to decrease bone resorption. So that means hold on to calcium in the bone. You want to decrease GI absorption of calcium. So that means if I ate something with a lot of calcium in it, go ahead and let a lot of that calcium go through. And then also you want to urinate, you want to decrease kidney reabsorption of calcium so you can urinate calcium out in the urine. And that's the story. Yeah.
What's the deal with calcitonin? Because the lab manual and the little table, it just said the effect is like unknown or
Huh? It says effect is unknown?
of calcitonin, like it has like some like insignificant effect.
Well, now you know that's not true. See, Miriam is going to lie to you a lot.
No, in
Separate.
physiological role in humans.
For calcitonin?
Yes.
I don't know. I don't know why Miriam would say this.
101.
But maybe she just got lazy and didn't want to write about calcitonin. Yeah.
Do we need to know the ranges for the exam, like of the blood calcium?
No.
Okay.
You just need to know that there is a homeostatic range.
Okay.
I won't ever ask you for numbers unless I specifically ask you for numbers. I'll put it in the color.
Okay.
Okay. From this diagram, what you should be able to do, so I didn't have anything in purple here, but now I've got instructions in purple for what you should do with this diagram. Start with the stimulus for a secretion of a hormone. So what does that mean in this diagram? Start, if you're studying this diagram, blood calcium. If you're hypercalcemic, yeah, or hypocalcemic. Know which cells respond to the stimulus. So if you're hypocalcemic, which cells are going to detect that? If you're hypocalcemic, going to be the chief cells. If you're hypercalcemic, parafollicular C cells. And then what hormones do they secrete in response to the stimulus? So parathyroid chief secrete PTH, parafollicular cells secrete calcitonin, and then know what the target cells are. The target cells are bone cells, GI cells, and kidney cells. So if you don't like my diagram, here's one from the book. You can look at that. Here's another one from the book that gives more. This is kind of cool, like if you're a chemist and you like to look at what vitamin D or calcitriol looks like or until it becomes calcitriol. So remember, it started synthesis of calcitriol or vitamin D in the skin, and then calcitriol became fully adult hormone under the presence of PTH. These are just flow charts. Oh, hey, come back. These are flow charts that talk about what we just talked about. So if you don't like my cartoon, you can study it in flow chart form, but we talked about that. And we've already talked about this, right? Hydrophilic and hydrophobic hormones. So we can go past this. Transport proteins. I like this cartoon because, so here's a transport protein. Here's a free hormone, so it didn't need a transport protein. So this free hormone can bind to the receptor in the plasma membrane, and then you get second messenger activation. And what's a classic second messenger that you guys drew with your GPCRs? G protein, it's the G protein coupled receptor pathway that leads to activation of a second messenger called
CAM.
cyclic AMP. Yeah, CAMP. So, just a little bit more detail on the transport proteins. Transport proteins allow the hormone to have a half-life. Who's heard of half-life? Half-life. Yeah, so the half-life just means the time it takes for the level of a hormone in the blood to decrease by 50%. So if you're a hormone and you have a half-life of an hour, then in 1 hour, 50% of you will be gone. That's what half-life means. They use it for radioactive particles too, half-lives.
So when does the transport protein release the hormone so it can pass through the cell?
Yeah, I think that's kind of a cool physics question. I think what's happening is the hormone is coming throughout the whole bloodstream. It's coming on and off and on and off the transport proteins really quickly. And then at some point, when it's near its target, the hormone will be off the transport protein and it will diffuse down its concentration gradient into the cell. So the transport proteins then increase the half-life of a hormone. So instead of this blue triangle hormone being gone in an hour, if it's bound to a transport protein, this blue triangle hormone might take 10 days before it's gone. Okay, talked about that, detail on that. We talked about that. Hormone cannot, hydrophilic lipophobic hormones, we talked about those. Now, here's the G protein coupled receptor pathway. This is in your book. So here's the plasma membrane, phospholipid bilayer. Here's the receptor, this big purple thing sticking in the membrane. Here's the G protein. So the hormone is the first messenger, binds to the receptor, G protein coupled receptor. That activates this protein called a G protein. When the G protein is activated, look what it can do. It can move in the phospholipid bilayer. It binds to an enzyme called adenylate cyclase. Adenylate cyclase cyclizes ATP. So it takes ATP and makes it into a circle called cyclic AMP. And then cyclic AMP can activate a protein kinase. What do you think a protein kinase is? It's got ACE on it, so it has to be an enzyme, yeah. So protein kinases are kind of a big deal. Have you heard of the human genome? Anyone heard of the human genome?
Yeah.
Tells us what our DNA is, basically. Well, there's a human kinome that lists all the protein kinases in the human body. There's just hundreds. So an active protein kinase can do a lot of things. And specifically, when you are an active protein kinase, you're an enzyme, you like to stick phosphates on things. That's the way Mother Nature designed it. If you're a molecule, you're sitting around, you don't have a phosphate on you, you're inactive. If you get a phosphate stuck on you, boom, you're active. That's just the way she designed it. And if we look at the periodic table, the elements over here, this phosphorus, right there. So in every cell of your body, you've got this phosphorus. Is that?
That's glorious.
Sorry, where's phosphorus?
K is that
I need new glasses.
phosphorus K. You don't have that from memory?
Please. I should. I'm not a chemist though. I've got parts of it memorized. Like I know where arsenic is because that's my dissertation. Oh, now I can see it. Yeah, here it is. Phosphorus. So phosphorus gets tricked out in the cell into phosphate. And then these protein kinases take phosphate and stick them on molecules, and molecules get activated that way. There's biochemistry 99. Would you agree with that? She's molecular and cellular biology, MCB. Easy way to put it. So now you're an active protein kinase. Now you can go do things. And if you do things, you're going to, for example, you can activate other enzymes. You can tell cells to secrete things. You can open an ion channel. Where have you heard that before? Opening 201, opening ion channels. Yeah, to do action potentials. So you don't have to know all the details of this. I tell you down here initially, so the first thing, the ability of this target cell, so this whole thing is a target cell, the ability of that cell to respond to a hormone depends on if the receptor is there or not. If there's no receptor, you can have a billion hormones out here trying to tell this cell to do something, but if there's no receptor, why is it that that hormone can't get inside the cell to tell it what to do? Why does this hormone have to bind a receptor? Why can't it just move right through the plasma membrane?
Hydrophilic.
Because this hormone is hydrophilic, aka, how's it feel about fats? Lipophobic. Yeah, okay, good. So ability of the target cell to respond to a hormone depends on the receptor. Once the hormone binds to the receptor, the ability to respond to that hormone depends on ICF biochemistry. What's ICF? Intracellular fluid biochemistry. So all I mean by that is if someone has a disease where they can't make a G protein, then even though the hormone bound to the receptor, but if that person can't make this G protein, you can't make the second messenger cyclic AMP, and you can't get the cell to do what you want it to do. I don't know if there's any diseases like that. That person would probably be dead if you couldn't make your G proteins. But so you have to have not only the hormone, but you have to have all this biochemistry and all the proteins in the biochemistry and all the enzymes. They all have to be working too, so the cell can do its job. So more purple stuff that you need to know. The G protein is going to activate the second messenger cyclic AMP using an enzyme. And then it says, what if this target cell used guanylate cyclase? Would it still form cyclic AMP? So what I'm saying is, instead of adenylate cyclase, you had guanylate cyclase. Could guanylate cyclase take ATP and make cyclic AMP?
No, it doesn't have a receptor.
It couldn't, but it could take not ATP, but GTP. Yeah, and make cyclic GMP. Do you remember where we talked about cyclic? You probably don't remember this. From 201. It was really hard. Cyclic GMP in rods and cones, the dark current. Well, does anybody remember cyclic GMP? No, okay, doesn't matter. But anyway, adenylate cyclase takes ATP and makes cyclic AMP. Cyclic AMP is the second messenger. That's going to be important because I'm going to ask you on the test to identify the second messengers. Okay. This is just another cartoon showing you the same thing. This is from the Saladin text. I like this cartoon because it shows all these hormones that use the G protein coupled receptor pathway. So adrenocorticotropic hormone, FSH, LH, PTH, we just talked about PTH, TSH, glucagon, we just talked about calcitonin, catecholamines, that's epinephrine and norepinephrine. All these hormones use G protein coupled receptor pathway. So this cartoon is not really showing you anything different, it just gives you the list of hormones that use the pathway. And here's all the steps that we kind of went over. So you can use this. And then this just tells you, well, for example, CRH, what's that? Corticotropin releasing hormone stimulates the synthesis and release of ACTH from what kind of cells in the anterior pituitary? Corticotropes. Yeah, nice. Good job. And then you have to turn the signal off, so this cell is not just going to make, it's not going to do its metabolic effects forever and ever. The pathway is turned off when cyclic AMP gets degraded, or the pathway turns off when there's no more hormone binding to the receptor. Okay. On the test, it's a concept of amplification in G protein coupled receptors. So all it means is amplification concept is you have a small stimulus, so you have one hormone is going to activate, I'm just making these numbers up for illustration. One hormone activates 10 G proteins. Each one of those 10 G proteins activates 100 adenylate cyclases. Each one of those 100 adenylate cyclases are making cyclic AMP. Cyclic AMP, a single cyclic AMP molecule can activate 1,000 inactive protein kinases. And then those active protein kinases can each activate 10,000 inactive enzymes, etc. So you get this amplification effect. So you have one hormone can give you a huge metabolic effect in the end. Yeah.
How does the G protein like move? Does it use GT?
Oh yeah, that's the detail, but yeah. GTP gets hydrolyzed to GDP. That means the phosphate burns off. When the phosphate burns off, it releases energy, and then that G protein can just kind of shuttle along the plasma membrane to get to adenylate cyclase. So again, phosphates are playing a role there. So amplification for the test, turning on a single hormone molecule, which results in the production of a whole bunch of product molecules down here. So you have a buttload of enzyme products that can give you these various metabolic effects. I kind of like this. I worked hard to make this movie for you guys, so I'm going to play it. Now, where did we put I think we had the sound level up here last time. I hope this doesn't blast you.
When a signal molecule, such as epinephrine, binds to a cell surface receptor protein, it activates a G protein on the inside of the cell. The G protein then stimulates adenylyl cyclase to produce large amounts of cyclic AMP from ATP within the cell. The cyclic AMP then binds to and activates a target protein, such as alpha kinase, which adds phosphates to specific proteins in the cell. The effect of this phosphorylation depends on the identity of the cell and the proteins that are phosphorylated.
All right, that was pretty good. And I think I'm going to stop here for now, and we're going to do some activities. Maybe we'll get to the end. So do you have your lab notebooks?
Yeah.
Okay, cool. I've got some, obviously, you can borrow.